All right, now it's time for part 3. What are we actually going to be doing with the SPR? The first thing I want to talk about is I want to show you some data that I collected with my last project, and then I'm going to move on to show you the data you're going to be collecting with this project. This is where we start to talk about the data and what it's going to look like. My first project was I was looking at a particular protein-protein interaction. I put one of them on the chip, and I flowed the other one past it through the flow cell. The proteins we were looking at were actually, when cells get stressed, they send out like a little white flag on their surface, a protein called MIC-A. And the immune cell has cells, the immune system has cells that are looking for this MIC-A protein. They have a receptor on their surface that will bind to the MIC-A protein. Okay? So when this receptor called NKG2D binds the MIC-A protein, the immune cell knows, hey, this cell is stressed out. And what it does with a stressed out cell is it kills it. So it activates their natural killer cells. The stressed out cell, it could be stressed out by just heat, or more likely is stressed out by becoming cancerous. So this is a cancer surveillance mechanism. When cancer stresses a cell, it causes a cell stress protein to be put on its surface, and that protein, MIC-A, binds the immune receptor NKG2D. It's a really good system for Bacterial because it's a simple one-to-one protein-protein interaction that results in a big immune effect. And so we were able to look at that in Bacterial and actually find out some cool things about it over time. These are what the two proteins look like. The NKG2D protein is orange and red on the top. The MIC-A protein is in yellow on the bottom. And the thing about the MIC-A protein is it looked like the MIC-A protein was kind of broken. It looked like it had a part that wasn't quite right, so we wanted to fix it. And so we used protein design programs to try to fix the MIC-A, to try to strengthen the NKG2D MIC-A interaction, to both understand the interaction and possibly to make something that could activate the immune system for our own purposes. We were right and we were wrong. Okay, the MIC-A protein looked broken, and it turns out that when we tried to fix it, it didn't work as well. But when we broke it a little more, it actually worked a little better. And so it surprised us, and we let it surprise us. We followed it where it went, and it was kind of a cool story at the end. That was the first of many design projects that we did on this. And basically, we were just messing around with the amino acids at the interface, trying things to change the interaction and hopefully to strengthen it. And so the kind of things that we saw. Let's say that here we put the MIC protein on the surface, we covalently bound that, and we flowed the NKG2D over the surface. And so when we did that with MIC-A wild type, we got this response. Actually, if you zoom in on it, it's a little better than it looks right here. It's not a very high response. It's kind of a weak interaction. It's got a KD of around micromolar. When we get into those units, we'll talk about how to calculate it. But basically, it's kind of weak. It does its job, but that's why we thought we could do better. So we found that a mutation that would let it stick better. And you can see that mutation is the yellow line right here that gives you a higher response. More stuff is binding. Turns out that it's also binding faster and falling off slower, which is all good stuff. Then we tried another mutation, and you see that this green line is much better. You're getting a much higher response. So we were really onto something. We were really happy to see this. We tried 2 mutations, and those were slightly better. You see that this one is binding about as much, but it's binding a little bit faster. You see how the line right here is curving up faster, and it's curving off slower. So we can analyze that, and at the end of the class, we will analyze that. For right now, we're just looking at how much binds, and we tried different things, and here's a MIC-A that did not work as well as we thought it would. Okay, cool. We'll just move on then. So one of the things that you do is you can do a plot of how high does the line get. What is the response at the end of the injection, the response right here. Okay, so how high does the line get? And remember, that's the equilibrium point. And when we do different injections of different concentrations of the same protein, if we inject more protein, we get more response. but how much more response do we get? That helps us measure what the equilibrium constant of binding is. And so the basic plot right here is a curved line. So once again, we have a curved line, but there's not too much calculus to it. What you have is as you have the concentration on the x-axis increasing, you have the response on the y-axis also increasing. And the amount that it increases by tells you what the equilibrium constant is. And you see right here we have the wild type in the middle, and as we got better binding, which actually had, like I said, it had worse design scores at first, but we just went with it, and so we flipped our design hypothesis, and it turns out that we had a good hypothesis, we just needed to flip it on its head. So long story short, we made mutations that caused higher binding, and you can see that they, as we injected the same amounts of NKG2D, we got more binding to the surface. We got higher amounts of binding. So that curve will give you the equilibrium big KD. There are actually 3 ways to measure affinities, because you can measure the KD at equilibrium. You can measure the KD from kinetics. You can measure the curve of the line up and the curve of the line down. Remember, each of those is associated with a kinetic constant, a K off and a K on. That is what I showed you before, and that's from having 1 K off and 1 K on. Sometimes you actually see a more complex curve than that, and sometimes you can see 2-step conformational change binding models, which is a more complex binding model, but you can sometimes justify that from the data and from other experiments. So that gives you 2 Ks for on and 2 Ks for off, and that gives you a more complex fit, but it also is another way to get to measure your KD. And we're going to do numbers 2 and 3 when we get to lab 5. We're just going to do number 1 right away, and so you might want, we'll talk about those more, and I'll show you how to do that when we get to that. And this is the 3 different ways of binding. You see the equilibrium, you're measuring different concentrations that have been overlaid on this graph, different concentrations and how high the response units go for each of those. And as you inject more protein, you see more response, a higher response. You can also measure how the lines curve, and you see right here that you're actually trying to measure the curved shape of the line, and that will also give you a big KD. And the thing is, these are different by a factor of 2, which is actually not that big of a difference on a logarithmic scale, which is what we're dealing with equilibrium constants. So for each protein, we want to do 4 to 6 injections of different concentrations of NKG2D, and we want to do those sets of injections 3 to 5 times. That's how many injections we'll be wanting everyone to try to do in the lab. So basically, if your data looks like this, notice how this has the shark fin shape. If your data looks like this after you've done all the blank subtractions that we tell you to do, then you have good data. The blank has to be a good blank, because there's not only the automatic blank that comes from flow cell 1, we also do blank injections of stuff that's just buffer, and then we manually subtract that out. You need to make sure that your blanks look good, because you need to be able to manually subtract out. Again, we'll talk about this in the lab when we get to it. But that was what we did, and I did that for about 10 years when I studied the NKG2D McA interaction. But now we have a new project, and the new project has to do with another protein-protein interaction, but this one actually is a bacterial interaction, and so we'll be using bacterial proteins. There's a bacterium that causes a new disease, and there's this headline, that we want you to know about this STD you've never heard of, a new sexually transmitted disease that is caused by a bacterium that adheres to the human body and causes disease. And this bacterium is called Mycoplasma genitalium. And you see right there, as it says, Mycoplasma genitalium is as much fun as it sounds. Well, we're trying to cure Mycoplasma genitalium by looking, and again, we're not using the whole mycoplasma. Don't worry about health things. We're just looking at the proteins. And we're just looking at purified proteins and how they stick to each other. But that's how the bacterium sticks to the body. So let's investigate that and let's try to solve this problem. So we're going to be looking at bacterial adhesion proteins. We're also going to be looking at how they bind to the proteins from the human body that they adhere to. And we might also look at antibodies. We have some questions to do with antibodies, though I'm not sure we're going to do that in this class this year. So here's information about Mycoplasma genitalium. It's Mycoplasma means it's really small. And it's actually a little parasitic organism. It has a really small genome. And it actually has the smallest genome of any species. And so in that genome, it encodes all these proteins, including it encodes the adhesion protein. Our collaborators have identified that, we have purified it, and we now want to study how that adhesion protein binds to other proteins. It's called the MGPB protein, and it's at the tip of these little things that are sticking off, waving off of the thing. It's a particular domain within the MGPB protein called the 4A domain. So we're going to call it the 4A domain. And it sticks to human extracellular matrix, which has a lot of different components in it, but we have identified which of those components it appears to bind. So what we have to test is our collaborators have made lots of different fragments of the adhesion domain. And so it's the 4A domain. And what they've done is they've made a lot of different truncations of it. So they will cut off the ends of it and try to see if we cut off this end, does binding go down a lot? And if we cut off this end, it doesn't care. The other thing they've done is they've taken particular amino acid residues and they've mutated some of them to remove the functional group, remove the charged group, to make it boring and neutral. And we found out that sometimes the protein doesn't care if you do that. For some residues, the protein really cares. And that tells us that that residue is important to binding. Hey, we better zero in on it. Maybe if we can block that residue, we can block binding. So we have all these fragments to test. We know that they bind two types of matrix glycoproteins, fibronectin and plasminogen. And so I'm going to give you a chip with either fibronectin or plasminogen on it, and you're going to inject 4A domain versions or mutants over that, and you're going to see how far the response goes up. That will be real data that we can actually use. So I don't know exactly what you're going to find either, but we're going to run enough controls that we'll be sure that whatever you're seeing, that it's real. So your mission is going to be a 3-week lab. Now it's all the way on 423, and so maybe you'll want to go back and listen to this part 3 before you get to it. But it's a 3-week lab to collect data. We're going to meet on 423. We're going to see the instrument. We're going to practice pre-concentrating stuff on it. And then we're going to have you schedule a time over the next 3 weeks, 2.5 really. Once you schedule a time, I will do the amine coupling. I'll give you a chip, and I'll assign you a C4A protein fragment. And you'll have to schedule, oh, and by the way, I'll take the time to couple my chip, and it'll take a few hours at least to measure binding. Probably overall about 4 to 6 hours, although you can do those in different chunks according to your schedule. You can leave the instrument on standby. We're going to share chips, and so it's just going to matter when you sign up on the schedule, because I'll be able to tell what chip to have in at what time. Okay, so it's just important to stick to the schedule as you have it once we set it up. During the 2 weeks after 423, we will not meet as a lab during those 2 weeks, because in exchange, I'll just ask you to find 6 hours. I'll give you 6 hours there, and I'll just ask you to find 6 hours on the instrument where you can do your injections. I've asked for you to have lab access 8 to 8 on Monday through Friday. Actually, it says Monday through Friday here. I believe I've asked for weekend access too. I will double confirm that when I go back. So in 514, when we do move on to our next lab, to lab 4, the report will be due, and that will be equilibrium fits, which will be the ones with the asymptotes, the ones that I showed you that curve up, but where you're fitting the end of the response only. And then in June, we'll do lab 5, where we'll take the same data, and we will do kinetic fits of the data. So we're going to do this in parts, and we're going to do the equilibrium part first, and we're going to take the same data. You'll be done collecting data, but you will process it a different way for Lab 5. That is going to be your mission. And so we will talk about that more as we go through. Thank you for listening to this. This actually is a little bit, it has a lot of concepts from later chapters. I've moved it up this year because I wanted to do this as an online lecture. And I also want it to be sort of the theme for the course. We're going to be talking about these things in chapters 3, 4, 6, and 7. So half of the chapters involve concepts we're going to be using with this BioCourse stuff. So as you go, you might want to revisit these slides, and some of these things might make more sense as you go. It will be on test 1. BioCourse stuff will be on test 1. And so you'll have lots of practice with it. And I think that those are usually questions that people like to choose from the list of questions. All right, so that is the BioCore lecture. Thanks for listening, and I will see you when I get back from my trip.